All right. Thank you, Christiana. And thanks everyone for being here. Um, and you might have noticed in the announcement that I work for the United States Department of Agriculture, Plant Protection and Quarantine in Nogales, Arizona. However, um, I am giving this talk under my own auspices, um, not as a spokesperson in any way for USDA or APHIS or Plant Protection Quarantine. And I'm going to be talking about activities that I've done generally on my own time and with my own resources. So for that reason, I'm not, and especially misadventures. I wouldn't want anyone getting the wrong idea that USDA is sending me out to do some of these things that might end me up in trouble. So as far as danger physically. So with that kind of preamble, um, first of all, I just have here kind of a collage of some of my favorite, most beautiful Mirids, just so that those of you who are maybe not so familiar with the family can see how varied they are in color, form, um, and also in size, although, these are all kind of shrunk or expanded to be about the same size. But anyway, it's a lovely group with a lot of beautiful species. Although a large number of them are also cryptic colored, so they would be kind of brown with various light colored mottlings, and they're beautiful in their own little way as well. Just a little introduction to me. So I got my start with, I guess, professionally as an entomologist when I was in high school, and a very thoughtful and observant freshman science teacher. When we went on a tour of a Western Cotton Research Laboratory in Phoenix, he noticed that I was asking a lot of questions of the entomologists there, and he ended up inquiring about an internship if they would have one, and or consider one, and they said, yeah, sure. And he said, helped me set it up, and I ended up working there for 4 summers. So that was my start. And then I went on to get a bachelor's in science in ecology evolutionary biology at University of Arizona, which I chose that of all the 3 state universities because it had the coolest looking mountains that were relatively near to the city. And Phoenix was too hot. That's where I'd grown up. But yeah, that was kind of my criteria. And then I went on to get a doctorate in entomology at the University of Kansas, after which I went into public service as a technician and later an officer in the Asian longhorn beetle eradication program, where I was on the shore of New Jersey and also working on Staten Island, which was an adventure all its own. I did not realize that there were a ton of blueberries growing on Staten Island, and it was kind of fun being able to pick and eat blueberries while working. Then an opportunity came up in Nogales, Arizona for me to apply to be an insect identifier with plant protection and quarantine. And I was able to get that position, and I've been here for 17 years now in Nogales, Arizona, which is right on the Mexican border. If you draw a line straight down from Phoenix to Mexico, that's pretty much where you'll hit, pretty much in the center of the southern border. And what my regular job entails is basically identifying insects that come across with cargo from Mexico, as well as insects intercepted with passenger baggage. But I also am very passionate about collecting insects, and I feel like it's just a real joy to look at them under the microscope, and to find new to me and interesting to me insects. And also, I really enjoy adventuring and going out and collecting. So, also, here's one form of arthropod diversity sampling that I'm interested in, which is the culinary form, also known as entomophagy. And up here in the upper left, there's me with, I believe it is a bark beetle pupa on my tongue while we're doing some peeling back bark and looking at bark beetles. And then here in the middle panels, there's a giant desert centipede, which are perfectly edible and quite delicious. And I cook that up, and there's me finishing it off in the bottom. And then the lower left is my son Nicholas when he and I did a few days of survival challenge, and our main protein during that time was the cicadas that we were able to shake out of the tree. So he has a skewer with some cicadas that we roasted on the left, and on the right is a rattlesnake that we found, and its head had been driven over by a car on our street. The rest of it was still good. So anyway, no otherwise healthy rattlesnakes were harmed in the making of those steaks. And then on the right here, usually during the summer, I will stock up a supply of cicadas, especially the dog day cicadas come out in August because they're rather large, and you can easily shake them out of the trees at night and collect them. I transformed these ones into some lovely oatmeal raisin cookies. Basically, just blended up the cicadas minus the wings and used them as an egg substitute. So anyway, if anyone ever wants to talk about eating bugs with me, I am totally game for that. So I'm going to tell you a little bit about mirids now, for those who may not be so familiar. They differ from other true bugs in the family Heteroptera. I mean, the order, suborder Heteroptera, with some key characteristics. And the image that I'm going to have in each of these slides that has a red border around it is a non-mirid that I'm just using for comparison. So a different kind of true bug, all right? So like many but not all other families of true bug, they have 4 segmented antennae. They have a single dorsal abdominal scent gland, which is under the wings here. They have a section of the wing called a cuneus, which is wedge-shaped or triangle-shaped, more or less. And some bugs have this kind of well-separated. I mean, some families have that well-separated and others don't. Most don't have it as well-separated as mirids do. And then mirids have kind of unique venation in having 1 or 2 membrane cells rather than branching or anastomosing or net-like kind of veins like you see in the oxycorinid in the bottom right there. Some other features of mirids is that they often have multiple types of CD. In this case, we have some long kind of flying CD like that. We have cool patterns of silvery, they call it sericeous or silk-like CD. And then it also has short scale-like CD in between those patches. Often I would say in almost all subfamilies of mirids, the ocelli are absent. There are no single simple eyes between the compound eyes. The exception would be the subfamily Isometapinae, which do have ocelli. So that's fun. And then for comparison, here is a geochorid, which has very simple cetacean. In fact, there hardly see any, but those that are there are just simple CD. And it has ocelli between the compound eyes. And my final characteristics slide. The males have asymmetrical genitalia. So as you can see, here I have the the whole insect habitus next to the close-up, so you can kind of appreciate what they look like. The right paramere in this top one is kind of elongate and maybe a little bit hockey stick shaped. The one on the left is C-shaped and kind of envelops the right paramere that kind of folds down onto it. Also, the actual genital capsule is asymmetrical in a lot of cases, often having some sort of tubercle on one side or the other. It's different than the other side. And then in this bottom left photo here, Phytochorus albidopictus, the right paramere is kind of triangular shaped with a little hook at the end. The left paramere is kind of U-shaped or S-shaped. And so for comparison, on the right, there's a Lygeid. And even though the bases of the two parameres are kind of offset from each other in the genital capsule, the parameres themselves are basically mirror images. And that's pretty typical in other families of Heteroptera. So I lifted this very helpful and lovely graph from Cassis and Shu. What this shows is accumulation of described species of mirids throughout the world, broken up into different geographic areas. And what we should know is that mirids are the largest family in Heteroptera with over a third of all the species. And as you can see from these graphs, if you were to add them all together, it's approaching 12,000 described species worldwide. And although some of these curves seem to be kind of leveling off a little bit, overall, I believe they give an indication that there's still a lot of new species to be found. Now, why aren't more people out there hunting for mirids and describing all these species, right? Well, turns out compared to most other insects, at least most other Heteroptera, and certainly pretty much all Coleoptera, these are very delicate specimens. They're difficult to work with. They have a habit of shedding appendages when you just look at them wrong. And so there's kind of a learning curve to being able to work with them in a way that preserves the specimens. But take heart, it can be done. I've been working on mirids really just since 2017. And when I go back and look at my first specimens that I pointed at back then, they are not very good. A lot of them are missing parts, but I could see steady improvement over the following couple of years. And now I always try to get them museum quality, even if I'm pointing up hundreds of them, because you never know where a specimen is going to end up and you want it to be good. So another thing that's both cool and also a little bit difficult is that mirids are usually associated with a particular type of plant. And that could be, in some cases, a species of mirid will be found on all sorts of plants within a family, like Asteraceae, or they may be restricted to just one genus or even just one species. Like there's some that are only found on one species of ambrosia, for example. And what this means is there's some really interesting biology to study there. It also means that because they don't all come to light, in a lot of cases, you actually have to go find the plant that they're on. And that plant may be in a unique habitat, like growing on a cliff or at the very tip top of a pine tree or on a salty playa or seashore or something like that. So it kind of invites people to go out on adventures, I believe, because you have to go out and find the plants that mirids are found on. So the collecting methods that I use, I know there's a lot of others, but these are the ones that I typically use. Night collecting with an ultraviolet light on a sheet. Sometimes I'll use mercury vapor lamp as well. But if I'm hiking around and stuff, then I'm just going to bring a battery and a UV light. Um, and then, so that can be really good, and you get a good variety, and that'll give you an indication of at least some of the species that are found in that area. Obviously, it doesn't give you a whole lot of information about their host, except that their host might be nearby, maybe. Although I find some that I'm like, holy cow, these are found on creosote bush, and there's not a creosote bush for like dozens of miles from here. So that's interesting. So you can get more information about the plants that species are found on by beating vegetation, and that's been my favorite way of collecting. Sometimes I'll use a beating net, but more often I use a simple white plastic wash basin, like you see here. And you can get one for less than $10, at your local big box store. So easy to replace if it cracks or something. Although I tend to tape mine up when they get cracked and keep using them until I really can't anymore. And what I like about them is that the insects tend to take a little longer to get out of them because they don't have the fabric to run along. And those steep walls just kind of keep them in there, especially at night. So I'm there with the headlamp. You shine that headlamp down in there and they don't really know what to do. So you can aspirate them at your leisure almost. And then I'm also I enjoy climbing trees. I don't do a lot of climbing big trees with ropes, but I did have a chance here with help from my friend James Korecki, who's an expert tree climber. He showed me how to get up into the top of a pine tree. And I found some interesting Heteroptera nomirids. I found some rhiparochromids and interesting scarab up there on that occasion. But that's just an example of a habitat that, not a lot of people go up to check that. And that would be an interesting place to beat insects out of those trees. Sometimes I'll just climb up from branch to branch and just kind of if I have a light running under the tree, and I'll just jump on the branches to try to shake stuff out of them and hope they go to the lights. So it's nice to have family support when you go on trips and you want to collect bugs when they want to do other things. As you can see, my sons here are very happy because Dad's taking a long time collecting bugs. Also, we went to a place in this case that was supposed to be called a lake, and it was really we were in the midst of a years-long drought, and the lake had dried up to just like a muddy puddle like this. But I do appreciate the patience of my lovely wife and my 4 sons who have accompanied me on many collecting trips, also known as family vacations. But I also go a lot on my own. And one thing that I'm really attracted to are the irresistible place names. So if you look on a map and you see that there's something called Silly Mountain, I mean, who doesn't want to write that on the label? This one's an outlier mountain of the Superstition Mountains range, east of Phoenix. And then there's me with my family this last year, where we posed in front of the Nothing Arizona sign. And that pretty much sums up what's there. But I did collect some bugs on just because I had to off of some of the shrubs growing around there. So another thing I'm interested in is some habitats that might be actually endangered habitats. And in this case, this is a remnant ponderosa pine forest in the Davis Mountains Preserve of the Nature Conservancy. I know Katrina has been there and done a lot of work collecting and describing new species from that area. And this is one of the few sky islands of Western Texas, or of Texas at all. A sky island meaning that it's a mountain range that is isolated from other high topography, where you end up with different kind of ecotones as you go up, and you can get all the way up in some cases to like an alpine habitat. but they're isolated by distance, and usually by expanses of desert or other, low-lying areas from other such islands, other mountains. So in this case, this is a relatively small mountain range, and it has been covered in, this pine forest, the upper reaches of it. But they had a big fire a few years back, and it destroyed a large portion of their ponderosa pine trees, as you can see by this kind of burnt out hulk of one in the upper right photo. But there are living pine trees too, so there are some of those further down in the canyon there, as you can see, and on the middle bottom of the upper left photo there. So this is a situation where, with more drought and with a couple more fires, or even one, that whole mountain range could lose all of its pine trees. And I would be interested in seeing if there are any endemic mirids, because that are on the pine trees. There are a lot of species of mirids that specifically live on pine trees, and I was able to get some interesting specimens off of those. And then one of my misadventures and adventures was sleeping in this little shelter. It's kind of a Adirondack shelter, open on one side. I'm looking at the back side of it here, called Dave's Shelter. It's up at kind of a pass between mountains there. And so I slept in there on the ground, in a sleeping bag, and at night, this giant rat hopped out and kept jumping onto my sleeping bag. And so that was a fight that I don't want to have, but I did have. All right. So another thing that I really like are nearly roadless expanses. So places where you really have to hike to get to, because what that tells me is that probably not many entomologists have gone there. And so this is I've been working on hiking the Arizona Trail in segments. And this is my son, William, who very kindly accompanied me on this hike. It was about 37 miles with no roads to connect you to any part. So there's no shortcut. And here in the middle of it, there's some just beautiful canyons. Here we are at Martinez Canyon, and a lot of very interesting vegetation in those lower desert slopes. All right. And then another way that I kind of decide where to go, besides just, off the beaten path, is targeting host plants. And so this species, I've been kind of trying to collect as many species of Megalosalus as possible. And Megalosalus nigrocaput is reported on Lycium pallidum, which is in the Solanaceae. It's a little kind of spiny little shrub with silvery green leaves. And I use a website, among others. I use SEINet because you can get a nice kind of distribution. Like it'll give you points where specimens of these plants have been collected and put into herbaria. So you can see that little triangle there on the map. Shows that's in the Gardner Canyon kind of area of the Santa Rita Mountains in southern Arizona, which is very nearby to where I live, just about half an hour, 40 minutes away. And so I hadn't ever found this insect on that plant, but going the right month to where the others had been collected, and then going to this area where that triangle was, I found this plant alongside the road, hit the plant, and sure enough, I got this bug. So pretty cool way to do a bit of a treasure hunt. Okay, so now I'm just going to quickly show you, because I'm running out of time, some of the locations where I've gone insect collecting. So in the Southwest, generally, you can see where it is, mostly Arizona. I've had a couple months where I've worked in New Mexico and had the weekends free, so I could go up into the mountains there. There's a close-up of Texas and New Mexico. Um, on the lower right there are the Davis Mountains, a really cool place. Um, and then, yeah, up into the uh, Gila River valleys and uh, mountains of New Mexico. Arizona, of course, that's my home state and that's where I live. So I've gotten around a lot there. I have a goal to visit every named mountain range in the state. So there's still a few to do. Um, California, in the last couple of years, I've been taking trips in the spring after the winter rains to try and go up and just see what I can get especially the kind of desert mountains, but also along the coast. So this is as far as I've made it up uh, in California, at least along the coast. So there's still a lot to do. So some of the misadventures that I've run into in the process of collecting bugs in these fun places. Um, well, at nighttime, I've learned that sometimes a washout of a road can be hidden by grass, by tall grass just growing there. And I found that out by driving from a collecting spot. I drove like 5 feet, and then the tires went completely down into the front tires into a washout, kind of a gully going across the road. And it was so deep that the tires were basically hanging in the air, and the truck was resting on the frame. There was no one there to help me out, so I just basically had to go around gathering rocks and building up a platform that I could put uh, whatever you call that, a tire, the thing that you pump it up with, the thing that you prop it up with, a jack. That's what you call it. Uh, to put a jack onto, raise the vehicle a little bit, and then build up the platform more, and do that by stages till I could finally drive out of there. Um, Also, I've learned that it's best to hike up steep gravelly roads before you try to drive them, because there's been a couple times when I've had to back out from a very steep road leading to a mine or something, um, and sometimes slide a little bit as I'm backing up. So that's scary. Um, many trails I've been on are just littered with rattlesnakes. Sometimes they're at shoulder height because you're kind of clambering over rocks and they're right there looking at you. Um, I've mentioned rats of unusual size, other rodents, things to watch out for. And then sometimes, like on one trip to California a couple years ago, there was a cold snap in April, and I was expecting to get a lot of bugs, but it was just super cold and really high winds, and made basically collecting at lights impossible. Um, Fortunately, I was still able to get some things by beating. And finally, the rewards of these adventures are finding lots of cool bugs. And I intended to get a whole gallery of bugs to show you guys, but my time's short anyway, and this one will have to stand in for them all. But this one was actually found like right in my yard, practically. Um, came to light, and later was able to, by beating various plants in the area, was able to find it on a cottonwood tree, as well as a nymph that looked like it belonged to it, probably. Um, and so this is, as far as I can tell, a new species in the conspicuous species group, Phytochorus. Just in my yard in Patagonia, I've collected at least 8 or 9 new species of Miridae, and in southern Arizona generally, especially by going to places like the Goldwater bombing range, bomb testing range, and other places where not many people go, um, uh, have been able to get some more undescribed species of Miridae. So I have a lot of work to do, and also, working together with other Miridae specialists to help document the fantastic diversity of this family. And also just it's wonderful to get out. I love Arizona, love the Southwest, and just the views are wonderful. So with that, I will say thank you very much for your attention, and I will be done.